What happens next? After the polypeptide chain is synthesized, it may undergo additional processes. For example, it may assume as a folded shape due to interactions between its amino acids. It may also bind with other polypeptides or with different types of molecules, such as lipids or carbohydrates. Many proteins travel to the Golgi apparatus within the cytoplasm to be modified for the specific job they will do. Now we'll talk to you about summary of all the things we have done or we have mentioned. First of all, the protein is the synthesis of the process in which cells make proteins. It occurs in two stages, transcription and translation. Second of all, transcription is the transfer genetic of instructions in DNA to mRNA in the nucleus. It includes three steps: initiation, elongation, and termination. After the mRNA is processed, it carries instructions into a ribosome of the cytoplasm. Then, the translation occurs at the ribosome, which consists of RNA and proteins. In translation, the instructions in mRNA are read and the tRNA brings the correct sequence of the amino acids to the ribosome. Then the RNA helps bonds between amino acids, producing a polypeptide chain. After the polypeptide chain is synthesized, it may undergo additional processing from the finished protein.
Processing mRNA in eukaryotes. The new mRNA is not ready yet for translation. At this stage, it is called pre-mRNA, and it must go through more processing before it leaves the nucleus as mature mRNA. The process may include splicing, editing, and polydenylation. These processes modify the mRNA in various ways. Such modifications allow a single gene to be more used to make more than one protein. Splicing removes introns from mRNA. Introns are regions that do not code from the protein, and the remaining mRNA consists only of regions called exons that do code for the protein.
Transcription. Transcription uses one of the two exposed DNA strands as a template. This strand is called the template strand. The RNA product is complementary to the template strand and is almost identical to the other DNA strand. Called the non-template or cutting strand. However, there is one important difference. In the newly made RNA, all of the C nucleotides are replaced with O nucleotides. The site on the DNA from which the first RNA nucleotide is transcribed is called the 1+1, one site of the initiation site.
Steps of transcription. Initiation is the beginning of transcription. It occurs when the enzyme RNA polymerases bind to a region of a gene called the promoter that signals the DNA to unwind so the enzyme can read the bases in one of DNA strands. The enzyme is ready to make a strand of RNA with a complementary sequence of bases. Elongation is the addition of nucleus to the RNA strand. Termination is the ending of transcription. The RNA strand is complete and is detached from DNA. Translation. Translation is the second part of the central dogma of molecular biology. RNA protein is the process in which the genetic code in RNA is read to make a protein. Translation is illustrated in the diagram below. After RNA leaves the nucleus, it moves to a ribosome, which consists of RNA and proteins. The ribosome reads a sequence of codons in RNA, and molecules of RNA brings amino acids to ribosomes is a correct sequence.